Our next professor actually can't make it today. She had a conflict, so I apologize for that. But Besa Xhabija sent me some remarks to talk about her student, Nicodemus Monear. I'm sorry I could not be able to introduce him today in person at Meeting of the Minds. Nicodemus is a native of Flint. He began his studies in 2016. He joined my laboratory in the fall of 2017 and was my very first student. So Besa is new to us. She's 2 years in here at UM-Flint. It's been a pleasure having him in my lab for the past couple of years. During this time, he's trained students, helped me build and manage my laboratory, has represented our lab and the Department of Chemistry and Biochemistry at a number of conferences. He's also been leading author in 3 major projects and 3 papers. This has been recognized by the department this year as Nick has recently received the Department of Chemistry and Biochemistry Student Researcher Award. Nick, would you please come up?
Hello, everyone. Bear with me just a moment while I get set up. All right. Again, my name is Nick Monear. I joined Dr. Besa's research lab late in 2017, so I've only been working with her for about a year and a half now. About 6 months of that was just planning. So in not too great a time, we've actually made pretty significant progress on 3 separate projects, which I'm going to share a little bit about those projects with you today, and also just my experience, how research has benefited me, and why I would recommend it to any undergraduate research, any undergraduate student. 2 of our projects, we've been looking at embryonic stem cells, mouse embryonic stem cells, specifically embryo toxicology and different toxicity effects to embryonic stem cells. So I'll do a little bit of background just on embryonic stem cells. They arise from the inner mast cell of the early blastocyst, which is very, very early on in embryonic development. Embryonic stem cells, once they get to that stage of development, they kind of have 2 paths that they can take. They can stay in self-renewal, where they can actually just maintain their status as embryonic stem cells just about indefinitely, as long as inhibitors are present to maintain that status, or they can differentiate, and once they've undergone that differentiation pathway, they can become pretty much every different tissue type in the body. So embryonic stem cells are a very, very critical point in development. So this is just a little bit more about those different pathways that I was mentioning. They can either stay in self-renewal again, move on and differentiate and become all these different cell types, and once they've undergone differentiation, they can form embryonic bodies, which is a little bit different. And you'll see some different inhibitors. We have here a kinase inhibitor, leukemia inhibition factor, and these inhibitors are what maintain that status in self-renewal. And in self-renewal, there are some genes that are expressed more prominently than they are in the differentiation phase, which is a big point of our research and some of the things that we were testing. So regarding embryonic stem cells and the toxicity effects, we looked at 2 different toxins or teratogens. One is lead, and what we wanted to do is we wanted to recreate the effects that would have been experienced during the Flint water crisis. Another, which I won't share the results with you today, we're actually going to be presenting that for the first time this summer, but it's a bisphenol, specifically fluorine-9 bisphenol, and the reason why that's significant is that it is a major component in the manufacture of plastics and different polymers. The reason why we're looking at this in relation to embryonic stem cells is different things like baby bottles. This particular additive is used in the manufacture of the plastics that are used to make baby bottles, and that additive can leach out into the liquids that are contained. So again, if you'd like to find out more about that, please catch up with us after June when we present that data for the first time. This year, we were looking at a morphological analysis for lead toxicity, and what we're seeing is that with exposure to lead, cells that should be maintained in self-renewal with the inhibitors present to keep that self-renewal status, we're seeing spontaneous differentiation. What you see in the bottom row is actually just alkaline phosphatase staining that correlates some of what we're seeing. The reason why that's significant is that alkaline phosphatase, the staining is more intense for embryonic stem cells when they're in self-renewal than once they undergo differentiation. And what we see is that when we expose embryonic stem cells to lead, that alkaline phosphatase staining becomes much, much less intense. And again, spontaneous differentiation, which we shouldn't see because, again, we're trying to maintain them in self-renewal. And you can see here, this is kind of what it looks like when they start to spontaneously differentiate. When they're in self-renewal, they like to form these nice dome-shaped colonies that you can kind of see light coming up around the edges, but once they've differentiated, they kind of flatten out and form these kind of elongated cells. And you can see here where they're circled that we actually see spontaneous differentiation, again, when they should be in self-renewal. So lead is causing an effect to this particular phase of development. We also looked at gene expression as it relates to self-renewal and differentiation of embryonic stem cells. And what we're seeing is these 4 transcription factors, which are highly expressed in self-renewal. When we start to see spontaneously differentiating stem cells, we start to see the expression of these transcription factors go up. Now, as far as exactly what that means, we're still kind of poking at that particular question, but I guess the most basic question we're asking, is lead producing an effect in the developmental cycle of embryonic stem cells? The answer is yes, and some of those results are shown here in pretty stark fashion. The embryonic bodies that I was mentioning, once they've differentiated and we change the culture conditions a little bit, embryonic stem cells will aggregate and start to form these embryonic bodies. And what we're seeing is that when we examine these embryonic bodies over a specified time interval, the exposure to lead is causing the formation of larger embryonic bodies over a shorter time interval. So it's accelerating this process, and while we don't know exactly what all the different outcomes that might have, in a developmental phase where things are happening at thousands of times the rate they are than they would be in you or I right now, the opportunity for a misevent in development is just that much greater. And again, we are seeing this process is accelerated by exposure to lead. Switching away from embryonic stem cells, another project that we did is related specifically to cancer cells. We were working with a breast cancer cell line. We're looking at a flavonoid called bakelin. And bakelin is found in naturally occurring in a number of different plant species. The most common one is just regular household thyme that you'll find in your spices cabinet, along with several others that are probably more well-known in traditional Chinese medicine. But when bakelin is ingested, it's metabolized in mammalian species by the addition of glucuronic acid and becomes bakelin, which is a metabolically active form. And what we're seeing is when we look at the cell cycle of these cancer cells, we're looking specifically, the phase of interest is the S phase of the cell cycle, because that's primarily where most of the DNA is replicated and produced from. And we're showing that with exposure to bakelin, the S phase is experiencing an arrest. So we're actually seeing a shorter S phase and thus less cancerous DNA being replicated during that time frame. And again, this is, as you can see up here, this is in a time frame, a time interval as short as 24 hours we're seeing these effects. So the effect of bakelin and its anti-tumorigenic properties is pretty stark. This is just a picture of some of the cultures that we fixed at 48 hours and stained with methylene blue staining. And as you can see, exposure to bakelin is causing a reduction in the number of cells in culture as with increasing concentrations of bakelin. Our control, you can see on the far left, and then even as low as 100 micromolar bakelin exposure at 48 hours, we're seeing far, far less cancer cells in culture. Now, I wanted to speed through a little bit of that just so I could talk to you guys a little bit about what this experience has meant to me and some of my takeaways. And a big one is just the idealism that comes with education and academics versus the realities that we actually see when we start to research and probe a question that's not well characterized in the classroom and with any academic endeavor. We're using a well-characterized issue to illustrate a point. Any issues that come up with that, there's a well-characterized solution. And the reality is that when we're researching something that is a novelty, we're kind of faced with very, very ambiguous results from time to time. And to know what that feels like, I think, is very important for anybody who's going, especially into a natural science field, because the likelihood that we'll see some results that we don't know exactly how to interpret, and the only source of interpretation is what we can gather. I think that's a irreplaceable experience, as opposed to, again, the well-characterized situations that we see in the classroom. Also, the exposure to analytical methods that I personally wouldn't have had an opportunity to use or have any experience with had I not joined a research endeavor. Specifically, some of the technologies that I've been exposed to. Qualitative or quantitative PCR, measuring the gene expression, like you were seeing just a few slides ago, measuring the gene expression for cancer cells just as they are in a native environment versus exposure to something like Biochemistry. Using technology like that allows us to quantify what type of effect we're seeing. That's a technology that I wouldn't have had an experience of using otherwise. And also exposure to just more practical considerations and concerns that, as a research investigator, they're burdened with, I suppose would be the best way to put that. Grant writing, proposal writing, coordinating with different institutions that have access to technology that we don't necessarily have access to. While that's something that's not very hard to wrap your head around necessarily, just seeing some of the challenges and hurdles that my primary investigator goes through just to make this experience for me possible and actually facilitate an endeavor that we ultimately might be able to publish and share with the rest of the world. Those are things that I would not have been able to really get a grasp on how they work and how much time is devoted to those things. And it is significant for anybody who's actually considering research as a career. And the biggest takeaway for me from all this is that we're one cog in a much, much bigger machine. We're probing some of these issues and hoping to get unambiguous results, and in most cases we've been very, very fortunate to do that. But without administrative specialists to implement policy to make some of these changes take place, I suppose, and medical professionals to actually use this information in their treatment options, our research would be very, very insignificant without all of those different components and avenues to actually share them. So that's kind of been my takeaway, is just that the work that we're doing is important and the questions that we're asking are going to be significant, but we're a part of a much, much larger, push to get answers to some of these things, and without, again, some of the people in those different professions and other researchers who are also just poking these questions but from different angles, it kind of minimizes our work without having all of those other pieces present. So that's ultimately what I wanted to share with you today. But with that in mind, the different cogs in that machine, we owe thanks to several different people just for allowing us to do our work, the different aspects of funding, the different labs that we've been able to work in to actually accomplish some of our work with the appropriate equipment. We owe thanks to them. So that's been my takeaway and why I would recommend research to just about anybody, because it gives a little bit more of a bird's eye view of what people go through to actually get answers to some of these questions and hopefully make some changes, like finding a cure for cancer and different things like that. So thank you.